Okay, so this is the cell membrane, and first we're going to talk about phospholipids, which are any lipid molecule that contain a phosphate group. Phospholipids are what make up the phospholipid bilayer of the cell membrane, and they are amphipathic, which means that they have both hydrophobic and hydrophilic qualities.
Okay, so each phospholipid is made up of two parts, which are the head and the tail. So the phospholipid head is made up of the phosphate group of the phospholipid, and the head is hydrophilic, meaning that it will dissolve in water, and this is also the polar side of the molecule. And then the other part is the phosphate or the phospholipid tail, and this is made up of like the fatty acids of the phospholipid, and this part is hydrophobic, meaning it won't dissolve in water, and this is the non-polar side.
Okay, so the cell membrane, so the phospholipids, when lined up, make the phospholipid bilayer. And so with phospholipid heads on the outside and then the tails on the inside, and then the layer allows for the cell membrane to be selectively permeable, meaning that only certain molecules can pass through the phospholipid bilayer to exit or enter the cell.
Okay, so next we have integral proteins. So in the phospholipid bilayer, there's two types of these. There's the protein pump and there's the protein channel. So protein pumps do what's called active transport, and they use ATP to pump larger molecules through the cell membrane, like in or out of the cell. And then over here is the cell, the protein channels, and they do what's called facilitated diffusion, and this does not require energy, and it usually used for smaller molecules to enter or exit the cell.
Okay, so the integral proteins are part of the transmembrane, which basically determines what is allowed to exit or exit the cell, and they're located all across the cell membrane. But then next we have peripheral proteins, and they are attached to the lipid bilayer, and they're found on the outside surface of the membrane. They support intracellular and extracellular shape of the membrane, as well as taking information from outside of the cell and sending it into the membrane for the cell. And they can act as enzymes to catalyze reactions and transfer small molecules through the cell membrane.
Okay, so next we have cholesterol, which is located all over the cell. Here's one example where it is. And then cholesterol provides stability to the cell membrane, and it does this by interacting with the phospholipid tails and other structures of the cell membrane. And then the next thing that we have is glycoprotein, which is located right here. And this enables cells to recognize if another cell is foreign or familiar, which is called cell-to-cell recognition. And in addition, glycoproteins help cells attach and bind to other cells, which is called cell adhesion. Okay, so now we're going to move on to the functions of the cell membrane. So first we have junctions. Junctions are created by junction proteins, and they create a strong bond between individual cells, between the cell membranes of cells. So there's three types of junctions. The first one is tight junctions, and these maintain the polarity of the cell, and they're involved with recruiting signal proteins. Another type is adherens junctions. These are sources of adhesion between cells, and these junctions are repeatedly assembled and disassembled, and they're formed by cadherins, catenins, and cytoskeletal adapter proteins. And then third, we have the desmosomes junctions, and then these are also sources of adhesion between cells, and then they do adhesive reactions that are done through desmosomal cadherins. Next, we have enzymes in the membrane. So first, there's the ICMT enzyme, and the function of this is it adds methyl groups to the proteins as they exit the cell, which enables the proteins to function. And then, so the enzymes in the cell membrane are connected to the phospholipid bilayer, and the only way that they can be removed from the phospholipid bilayer is if they are dissolved. And then another thing about enzymes is that peripheral proteins can also act as enzymes in the cell membrane, and they can transfer information about extracellular activities around the cell.
Okay, so next is transport in and out of the cell through the cell membrane. And so this is done in different processes. First is diffusion. So this is when the movement of small molecules go through the selectively permeable membrane from an area of high concentration to low concentration. And so basically diffusion is just when molecules go through the phospholipid bilayer. And then we have facilitated diffusion, which is diffusion with the assistance of a protein channel, but does not require energy. So molecules are too large to just pass through the phospholipid bilayer, and therefore require just a little channel they can pass through, which is the protein channel. And then next is active transport, which is larger molecules that are moving from high to low concentration, and they require energy to pass through the membrane. So the molecules will travel through the protein pump, in which ATP is required to pump the molecule through the membrane and into the cell for use.
Okay, next we have endocytosis. So this is when a molecule is too large to go through a protein pump, and then a vesicle will then be formed by the membrane surrounding the molecule, and which will transport it into the cell. And then the opposite of this is exocytosis, and this is just when a large molecule needs to exit the cell, and then so the cell membrane will engulf the cell and then transport it to the extracellular side of the plasma membrane, and that will release it out of the cell.
Another function of the cell membrane is recognition. So this uses the glycoproteins found on top of the cell membrane to allow cell-to-cell recognition, in which a cell can realize if a cell is familiar or unfamiliar, and as a result decide to bind or not bind with the cell. Peripheral proteins can also be involved because they take in information about the environment outside of the cell and report back to the cell membrane and the cell through the use of the proteins to determine if binding is a possible option with another close cell.
Okay, so next we have anchorage. Anchorage, or it's also called cell adhesion, is when two cells bond with one another or another surface in order to survive and divide. Through connecting with proteins on the cell membrane, cells can stick to one another or to a surface in order to control division and ensure the cells stay alive.
Okay, lastly is signal transduction. So this is when a receptor protein receives messages from an outside molecule and then chemically binds together through ligands, and then this changes the receptor in some way and initiates transduction. Protein transduction relies on the loss and gain of phosphate groups or the creation of ATP and ADP, and when the phosphate group is lost, transduction can begin through the transduction pathway. This is regulated by the cell to ensure that there aren't too many molecules entering the cell, and the molecule ultimately conveys a message to the cell in which the cell changes function to combat some extracellular or cellular activity, and these can include regulation of gene expression, regulating of certain proteins, cell division, or even apoptosis.